and my perspective of the United States has been California. So you can imagine it's a little bit of a skewed perspective. But I have only lived in California, and um, just on Wednesday, I took my citizenship test. So now I'm going to become a citizen. So I've lived 20 years in California. And then, thank you. We are very excited about it. We've so I've lived basically 20 years of my life in India and 20 years, I mean, I'm a little over 40, but 20 years in California. But they say the first few years of your life, you don't remember, right? There is another fun fact about me. I was actually born in Santa Cruz.
What?
No, not Santa Cruz, California, but in Mumbai, where I was born, there's a suburb called Santa Cruz. My parents still live in Santa Cruz. So my home address in Mumbai is Santa Cruz East, Mumbai. So it's kind of funny that, after all of this, I have come back to Santa Cruz. And so my birth certificate also has born in like Santa Cruz, Bombay. Yeah. Yeah. But I've kept this. I study cancer. Primarily, my lab is focused on breast cancer, and I'll, talk a lot about breast cancer. But I did my PhD in colon cancer. So my initial, all my studies have been during my PhD was focused on colon cancer, and then during my postdoc, I've switched to studying breast cancer. And I actually say I'm not a cancer biologist. I'm a developmental biologist in some way, or a stem cell biologist. So I look at cancer from a very developmental perspective, and I try to see like what are the things that in our normal body that are functioning that go wrong in cancer. So that's why there is stem cells, and that's why I look at the mammary gland, which is what we call the breast in the mouse. We use the mice a lot in the lab. So I'm just going to dive in, but I think there's going to be a lot of information, and I don't know if there's how much jargon is there. So please stop me at any time, and I hope this is more a conversation than me standing here and just chatting and you folks just ask questions. So why do we care about, besides the breast, why do we care about breast biology? So there is just some statistics beyond, how do I hide this? I think I can.
I think you can slide it up.
Oh, I can. Maybe here. Okay. So why should we study? So I think my animations are not working. Let's see, maybe I have to do this. Okay. So actually the statistics are that 1 in 8 women in their lifetime will have a diagnosis of breast cancer somewhere during life. And that's a pretty big number if you just, think about the percentage. And this year, about 43,000 women will die of breast cancer in the United States. In fact, this number is more than, people, soldiers that are lost sometimes at war. And then, I do not like this, but this is going to be hard if I have to, but we sometimes forget that as a biologist, we forget that the primary role of the mammary gland in all mammals is to, this might be better. Is to produce milk for the infant, right? That's the goal. And in fact, there is a large number of women that experience lactation insufficiency. We don't talk about this enough. We always talk about breast cancer, but and we are told, even I became a mom, yeah, you're going to put that baby on there and it's going to feed, and no, it doesn't work out that way. There's like so much practice that went into, and there are a significant amount of women who actually experience lactation insufficiency, which can contribute to a lot of other things. So I think studying breast biology, yes, cancer is important, and that's where I'm focused, but I think there is also this whole idea of like, just how does this milk production happen and how is it so controlled and what can we do? Also because we have dairy farming in the United States, which is one of the largest industries. And if we can find ways to increase milk production, we will be able to, in fact, change the way our environment is as well, because 1% of US GDP each year, that's the dairy farming industry. One cow produces greenhouse gas equal to one car driving for 7,500 miles. So this is not just, understanding, like I'm just trying to put everything that is there about why we should be studying the mammary gland. Um, Okay, so I'm going to just talk a little bit about what, the gland looks like. Because we're going to talk a lot about the cells that are in the gland, which ones produce milk, which ones do other things, and why they are there. So if you folks want to interrupt me at any point in time, please do. So this is just the structure of the human mammary gland. And this is the structure of the mouse mammary gland. So the gland over here actually is this, uh, is this pink thing. And this, and the one that is like a tree over here, that is the gland. The remainder of all of this is in fact fat. So the way it, and we'll get into like how this happens during puberty, but most of the milk in humans is produced in this structure on the top, which we call TDLU, or terminal ductal lobular units, just a very big name. But that's where the milk is produced, and then it gets pushed out to the nipple to feed the child. The mouse mammary gland, which we use very commonly in the lab, because we need to have a model system to study this, and we can't study women. That, I mean, there are some women who donate a lot of their tissue, or even donate milk so that people can do studies with human samples. But most of our work is done in the mice. So it's a little different, but in basically it will, the structure is like a tree, but the tree structure is a different. So just, but the organization inside is the same. So this is what it looks like. So if you take that same structure, basically it's like a tree-like structure like this. And then if you cut a section across this tree-like structure, what you see are these two types of cells. These cells on the inside, they are surrounding this hollow tube. And usually in biology, whenever there is a tube in anything like our intestine, or even the lung, we call that a lumen. Lumen basically means the hollow part of the tube. And so the cells that are next to the lumen, so each of these is an individual cell. We call them the luminal cells because they face the lumen on the outside, they are inside. And the cells that are actually on the outside, we have this terminology for anything that is not facing the lumen. We call them basal. It's just the terminology in every, even in our skin, we call like something facing the outside, and the the basal part of the skin where the skin grows, we call them the basement membrane. That's what, it's a cell biology term. So these cells on the outside, we call them basal cells. And I think it's, we can actually stain these different cells. So this is basically a mouse mammary gland, and these green cells on the inside, each of these green round things is one cell. And those cells on the inside are the luminal cells, and the red cells on the outside are the basal cells. So this is what a mouse mammary gland, or even a human mammary gland, if we just can take a small thin section over here and look at it, that's what it would look like. Any questions? Think. Okay, so this is, something that pathologists often, when folks go in, and I don't know if someone has heard the term biopsy, but when they do a biopsy of something, what they are doing is they are taking a small piece of tissue and they are putting it on a slide, and then we can look under the microscope to see what kind of cells are in that. So that's what we do with the mice. That's what we do with any of the human samples that we get also. Yeah. Okay. All right. So yeah, the mammary gland, with whether it's the human breast or from a cat, dog, or even mice, which is what we use in the lab, it is very unique and different from any structure in the body. The reason it's very unique is that actually most of the development occurs during puberty. So interesting fun fact is that men and women actually, when they are born, they both have a rudimentary ductal tree, or they have that small tree that is there, which is not developed. So this is just showing a mouse mammary gland. So I'm just going to take you through it. So this is the dark pink structure that is over here. That is actually the tree, and that's how it is even in humans. It's very tiny near the nipple, and then most of the development will occur during puberty. Now mice have a very live for a short time, so they live for 2.5 years. We live for like 100 years now is the average. So like our pubertal development occurs between 13 and 18 years. Mice finish their puberty at around 12 weeks. So their pubertal development occurs between 4 weeks, around 3 weeks to 10 weeks. That's when they undergo puberty. And so what we can do is take the glands from the mice we have mice in the lab at different times, and we can take the glands from the mice and put them on a slide and look at them and see how they are growing. Does that make sense? Yeah. Okay. And so then we can stain them, and then this tree-like structure, it will slowly invade the fatty tissue that is there to actually form this really dark, it will fill up this entire gland with the tree-like structure. So this is in response to estrogen that is being produced during puberty. And in fact, so males don't actually undergo puberty with estrogen, but they undergo testosterone. But if you take a male mammary gland and put it and give it estrogen, it's going to respond in the exact same way. So men also get breast cancer, but it's a very, very small, very tiny amount of percentage of people who males get breast cancer. So it's not just, the tree like that initial tissue is there both in men and women. It just responds differently based on the hormone that it is seeing. And then the real cool thing happens during pregnancy is like during pregnancy, this gland will see that, there's progesterone going on in the system. So it's going to divide a lot more. These cells are going to just become more and more cells, and they are going to prepare for lactation. That's when the milk is produced, and then the milk is going to be released in that lumen. I have a picture later on. Yeah, I know that's something else. But basically, maybe here. So these cells that are over here, they are the ones that will produce the milk. So they are the luminal cells. They are going to divide a lot more to become the milk producing cells. And the milk is going to be released into this lumen inside. And then the cells that are here on the outside, they are the ones that are going to help in pushing the milk out. So when the baby is suckling, the oxytocin that is being released by the brain is going to help in contracting this so that the milk gets pushed out. Cool. Yeah, it's pretty cool. I think it's pretty cool. Okay. So then the other thing that happens with the mammary gland is that when this whole cycle is done, all these cells that are there in the mammary gland, they will die. And the mammary gland will return to a pre-pregnancy state. We call it pre-pregnancy. When I had my first kid, my doctor told me after she's like, remember, you're never going to go, your body is never going to go back to being exactly what it was before you had a kid. This is the new normal. And so even though these cells die, they the gland has some sort of a memory that it has been through a pregnancy, which is why they say, oh, second time around, lactation is so much easier. Because these cells have been through this one cycle, they remember it. But the next time this happens, and doesn't matter the number of times it happens, each time that cell divides a lot, and becomes this milk producing cell, and then again dies, and can do this over and over again, which makes you think that there must be some cell in the mammary gland that is able to do this multiple times. And that's what we call stem cells, basically. So have folks heard about stem cells?
Yes.
Okay, so can someone tell me, like, what I want to know, like, what kind of stem cells have you heard of?
Pluripotent stem cells.
Pluripotent stem cells. Okay. So, what does it mean to be pluripotent?
You can turn into any kind of cell.
Yes, yes, you can turn into any other kinds of stem cells that folks have heard of, that are there in all our bodies right now. Fetal stem cells. So fetal stem cells and pluripotent stem cells, they are the cells that give rise to all our tissues in the body. But the cells that I study in the lab are not those cells. So and we'll talk about this, but I think I should have put that slide first. But one of the things about every cell in our body that it is slightly different from each other. And I put the picture of M&Ms and jelly beans over here. Hopefully, at some point, you folks have tried either M&Ms or jelly beans. If not, I can run up to my lab and get some. But so what do you think is different about M&Ms and jelly beans?
What they're made out of. Materials they're made out of.
So, okay, yeah, that's materials. But in terms of like the taste,
Oh, that was chocolate. Yeah.
the one has, and so between, okay, maybe, and that's right. And then within the M&Ms, they all look different, but do they all taste different?
No,
No.
no, they taste the same.
They taste the same. And what about jelly beans?
They all taste different.
Exactly. So I think like the thing about our cells are that, some cells look different, but they are the same. But some cells they can be the same. I won't say they are the same, but we say maybe we don't know that, right? But some cells can look different and they are different. And I think this is very, it's a very important thing that we need to know, because most of the time when people did, when we didn't have the technology that we have today, most of the time people, when they took a tissue, what we would do is we would study the whole tissue, and we would not be able to study individual cells, which is why we didn't know a lot about stem cells. I think my next slide is the stem cells. So what are these stem cells? And I think somebody here said that, there are these stem cells that can become anything in the body. So we have like, we can have these stem cells which will make more stem cells. And then we have these stem cells which can become either the muscle cell, it can become blood cells, it can become neurons, and those are what we call the pluripotent or the fetal cells, because in the end, we all came from one cell. And I think that is so cool, right? We have trillions of cells in our body, but we all came from that one single cell in the beginning. But interestingly, in all our bodies, we still have stem cells, which are needed for the maintenance of our body on an everyday purpose. Like there are the bone marrow cells. So the bone marrow cells contain these stem cells, the blood stem cells, which is why some people may undergo bone marrow transplantation, right? Because if they have a certain disease, what they do is they try to remove those disease cells and give them new cells from a different person, so that the whole blood system comes from these new stem cells which don't have the disease. So, we have these cells which are the fetal stem cells. We call them embryonic or pluripotent, as you said. And then you have these adult stem cells. This is just one example, which they're showing blood stem cells, which can give rise to our white blood cells, the red blood cells in our blood, right? But just like the bone marrow stem cells, almost every tissue in our body has stem cells. Like the skin has stem cells, which is why when we get a cut, the skin can heal because these stem cells, they get activated and then they can come and close that wound. Our gut has stem cells. In fact, all of us get a new gut every 7 days. So every 7 days, if there is something brand new in us, it's our gut, because there are stem cells in the bottom of the gut, and as we eat and do our daily thing, a lot of the cells of our gut are being washed out when we poop. And so every time there's a new stem cell that is coming up in the gut. So that's another place that we have stem cells in the body. Then we have stem cells in our hair. So our hair follicle stem cells are there, which give rise to, we cut our hair, we have constant growth happening. We have actually, this is a little debated, but people are now showing that there is stem cells even in the brain. So people initially thought once the brain develops, it's done. But we know that now there are stem cells in the brain which are giving rise, that are involved in certain cases. They can be activated to respond to injury or something. And so almost every tissue, I think the lung has stem cells. I'm trying to think other tissues. Muscles have stem cells, which is why people can go to the gym and build new muscles. And, when bones have stem cells, which is why when we get a fracture, our bones can heal. So lots of stem cells, and these are not embryonic. We have a term for them. We call them somatic stem cells. So somatic just is a fancy way of saying they are the stem cells that are there in the adult body that are not going to give rise to the next generation, but they are there in our body to just help us live through 100 years of our life. So yeah.
So are these two kinds, the fetal, they're just different names for the same thing? There's just one kind of stem cell found in different locations?
No, each location has a different stem cell. So once the embryo develops, so the first cell that comes is like from mom and dad together, and we have that first cell, right? Then that cell is going to divide many times and give rise to all the different tissues in the embryo. But then each tissue is going to have its own stem cell that is going to be specialized for its own. So the bone marrow stem cells are going to make blood. The stem cells in the brain are going to make neurons. The stem cells in the gut are going to make intestine. And just like that, we have stem cells in the mammary gland, which are going to divide every time there is a pregnancy and give rise to the milk producing cells. And then those cells, the milk producing cells will die, but the stem cells will remain. And then this can happen again. When those stem cells see progesterone, they're going to divide again and then give rise to the milk producing cell. And then those cells are going to die. So the stem cells, that's why they are super important, right? Because they have this capability to divide and make more of themselves. And we have a very fancy term that I did not put on the slide, but it's called self-renew, which basically means you are making more of yourself without changing anything. And then they not only can make more of themselves, but they can make more of the milk producing cells. In my case, that's what I study. Or they can make more of, the cells that are needed if they are in the bone marrow, then they can make more of the cells that are needed to produce the blood for infection or something like that. Okay, any questions?
My quick question is that, so the milk producing stem cells get reactivated due to a specific hormone?
Yes, so they can see progesterone, and they get activated when there is a pregnancy. And and so interestingly, studies have shown that even if we inject progesterone into mice, we can activate the proliferation of these stem cells because they see the same hormone. And yeah, kind of cool.
This is a nerdy question. Does the stem cells have a better way of restoring their telomeres?
Yes, they do. They absolutely do. So I don't know if everyone knows telomeres here. Okay, so did Dr. Greider speak with them?
She actually spoke to the group, right, it was our last talk before COVID.
Okay. Okay, yes. So she must have said that how these telomeres are required, right? So, and not our somatic cells don't have activation of telomerase, which is what she's found and won the Nobel Prize for. But our stem cells have activation of telomerase because they are going to stay in our body for such a long time and be able to divide so many times so they have activated telomerase. But as they become specialized, they lose that telomerase. So then their telomeres are no longer maintained, but the stem cells maintain their telomeres. Yeah.
For those of us who were not here for that talk, what you're saying
Well, what is that?
Yeah,
Yeah.
telomeres. So what happens is our chromosomes, right? Our chromosomes actually, so we have, this will go a little bit into DNA replication, which will be, so I'm just going to draw I teach telomeres to my, so I have slides too, which I can pull up. But the way our DNA replication works is that we have the chromosomes, but there's a problem at the end. So we have like DNA. So we have our DNA is double stranded. So we have A, T, G, C, and A, T. I'm just going to say A, T, G, C. I don't remember the sequence of. And then we have on this side, we'll have T, A, C, G, right? And then we have this strand each time that our cell divides, the exact thing has to be copied. But the problem that happens in a very simplified way is the way the protein that is copying this, it cannot somehow copy the ends. So it's not made to copy the ends correctly. So each time that our cell divides, we get our chromosomes become a little shorter, which is a bad thing because with every generation, then the humans are going to get lesser and lesser chromosomes, right? So to solve that problem is where telomerase comes in. And telomerase basically is able to extend these chromosomes in a different way so that every generation has the same amount of DNA. So our kids have the same amount of DNA that we have, and over how many ever years humans have been around, the same amount of DNA is maintained. But we don't need to do that for all the cells in the body, right? Because if my skin over here, I would love that it would remain young forever. But if it doesn't have the little bit of DNA, it doesn't care because my children have got the full length of the DNA where my cells had this telomerase protein. But now if we have some stem cells in the body, they also need to have the DNA proper because if they don't, then they are going to get shorter with each time, and then we are going to age very fast. So there is a mouse that they made. Actually, I think Carol Greider's lab only made the mouse, which basically when they don't have this gene telomerase, those mice out of 5 generations, their chromosomes get shorter and shorter, and then they age faster. There is this movie on Netflix called The Age of Adeline. It's a fun romantic movie to watch, very, very chill. But basically over there, they show that she doesn't age. So she's 124 years old, but she looks like a 27-year-old. And they show that she has activated this gene telomerase. And yeah, it doesn't really work because the problem, if you think about it, is that if you have these stem cells that have always activated, we are going to come into the problem of cancer. Is that you don't want cells in the body constantly dividing also, because that's why it's very, very tightly regulated, and you have only those stem cells that have the telomerase that is active, so that they are going to give rise to the specialized cells which should do their work and then go away, and then the stem cells can make new and then do their work and go away. So now you can think about, like, based on what Mark said, is that what happens if they don't go away, right? Or some problem happens in the stem cells. And that's actually what I study is if those problems happen in the stem cells, how does that give rise to cancer?
Would you mind just spelling the word?
Yes, yes.
Because I'm not seeing it in my head yet.
Yeah.
That's a new one for me.
So and then this is the protein that that is needed for this end of replicating the ends of the chromosomes. And the ends of the chromosomes, they are basically called telomeres. That is the DNA which this protein can copy it. And so this is the problem mostly in all, like, humans and all the animals, but it's not a problem in bacteria. And because the way their DNA is different. So our DNA is like straight, long chromosomes. We have very large chromosomes. But the bacterial DNA is round. So they don't have a break anywhere. So it doesn't, so they don't have telomerase, or they don't have the same problem. Yeah. And yeah, and we have a lot of DNA. I mean, we have 3 into 10 to the 9 base pairs of, that's how much when I say base pairs, I mean one of this. That's how many we have in our cell. And because we get one set of chromosomes from our mom and one chromosome set, this becomes 6 into 10 to the, 3, yeah, I think that replication 6 into 10 to the 9. I might be wrong with the numbers because I'm really not good with numbers, but it's somewhere in the billion range. And then every time a cell divides, this is the amazing, that's why I like studying cancer. I mean, cancer is really bad, it's horrible. But if you actually think about it, every time a cell divides, it has to copy exactly all of that. It's like going to your car that many times, and every time the car has to start, that would be an amazing car or an amazing cell phone. Like Apple would have made the best computers ever if every time it did. So our cells have to do this over and over again, and we have trillions of cells in our body. So sometimes the cell can make a mistake, and that mistake can be in a place which might be very important for the cell. And this actually leads a little bit away from what I'm going to talk about, but still, like, and that will happen at some point in time in our lives. I think everybody has a cell which has not the correct form that is floating around in our body. That does not mean that we all get cancer, but just over the period of time, that one cell that got that one change can stay, and then maybe in the future it might get a second change that might stay, may get a third change, fourth, fifth. It needs, for becoming a cell, becoming cancer requires a it requires at least 5 to 7 changes, not just anywhere, in key places, which is, if you think about it in terms of probability, it's a very, very low probability. But because we just have so many cells in our body and we are living longer, and we have that probability increases. So that's why we hear that, cancer rates are increasing, but there's also, it's a numbers game to a certain extent. Does that make sense? Yeah. Yeah. Okay. I should have put some slides on just cancer also, but yeah. So this is what we then look at, right? We have these stem cells And you can ignore what this is because this is from one of my talks and I didn't have a way to cut it out nicely enough. But basically these stem cells can divide and they can give rise to these luminal cells and these basal cells, and they can do it over and over again with each pregnancy. And then over time, you can imagine that if a woman or a mouse goes through multiple pregnancies, what might happen to those stem cells? Will they have more stem cells or less stem cells?
Like they lose some.
They lose some, yeah. So and towards the end of the talk, maybe we'll get into like what happens with and without pregnancy to these stem cells and how that affects breast cancer risk. I think but we are still studying that. That's an ongoing project by a graduate student in the lab. Yes.
So if the typical birth control pills stimulate the breast to go through this process, as I understand it, it imitates the beginnings of a pregnancy. How does that affect the breast? If
So
you're taking birth control pills for a long time.
I think from what I know, there was this one study done like almost 20 years ago. It has a protective effect towards breast cancer in the end. So it stimulates it, but over time, because these cells then are decreased, so overall, the risk of breast cancer is less on birth control.
There's progesterone in the birth control pills.
Depends on which, yeah, little bit, yeah. But it doesn't go through like, there is some evidence that it can have a protective effect. But the evidence is minimal to say. There are other things that can have protective effects, like pregnancy has a protective effect against breast cancer, but the pregnancy has to be below the age of 30 to get the protective effect. But then here is the other thing. With pregnancy, there is an increased initial risk of getting breast cancer in the first 10 years. So the protective effect only comes after the first 10 years of childbirth. So breast cancer is super complicated. I mean, we put it all in like one thing, but actually there is so much other things. And age of the first menarche and menopause can also affect the breast cancer. So how long is the body exposed to estrogen can also have a effect on breast cancer. And it is very complicated. It is very complicated. It's like, this very, very big puzzle. And we have so many breast cancer researchers who are trying to just solve pieces of the puzzle. And somehow, by everyone's collective work, we can put like something together. And then we just find this other thing that we don't know about and we start doing research on that. So, okay, this is, so, I'm going to now get into a little bit of how we actually do all this stuff in the lab, right? So this I put this picture, and I know it's very complicated, but I'll take you to it. But I put this picture in because this was actually developed in Santa Cruz by Dr. Charles Daniels. He's like a pioneer in the mammary gland field, and he did a lot of work. I don't know if this building was there at that time, but so you did a lot of the work in breast biology and studying how these normal mammary gland grows over here. So everybody in the mammary gland field knows him. So when I go to any conference and I say, I'm at Santa Cruz, and he's like, oh, you're at the home of where all of this was invented. So that's really cool. So what we can do in the mouse is, we can, before the mouse undergoes puberty, we can actually go and do a surgery and take off this small tree of the mammary gland. We can remove it. And so now the mouse does not have the mammary tree. And then what we can do is take another mouse, and we call that the donor mouse. And so we can take the tree of that donor mouse and put it into this other mouse, which is 3 weeks old. It's this tiny. So the surgery is hard but doable. And then you can take the cells of those mouse and put it into this donor, from the donor mouse, and put it into this other mouse, which we remove the tree from, which we call the recipient mouse. And then we can see whether the tree grows or not. And this is what we call a transplantation assay. So this is the same thing as doing transplants in humans, but it's easier in mice because one, the way our mice are, that they all are genetically the same. So they have been inbred, also they have been breeding for like, I think 20 or 30 generations to each other. So they have the same genetics. So we can put tissue from one mouse to another mouse without having the other mouse rejected. This cannot be done in humans because, we all have our immune systems. And so if we take tissue from someone else into our body, we'll reject it, right? But the mice, because they are the same, we can do this. So it provides a really great way for us to understand these stem cells, because what we can do is we can take different parts. We can take either the cells that make the milk or the cells that push the milk out, and we can put different parts into the recipient mouse and see which one can give rise to the tree. And so then we can take this recipient mouse and make, a whole tree. We can also make the recipient mouse undergo pregnancy and see if the cells that we put, can they give rise to the milk producing cells. So lots of cool stuff can be done with the mice.
So you're trying to search for where the stem cells are located in the original structure?
Correct. Yes. So we are trying to look, I mean, people have tried to look, and we know now where the stem cells are located in the original structure. That as scientists, we always argue about exactly the location. So I just had an argument yesterday with someone about this, not an argument, but discussion. That's more, discussion is the right word, about where the stem cells are. And there are lots of theories like, is the cell always there? Or is it going to be activated only at some point or not?
Is this applicable to humans?
We can't do this in humans. We only do this in mice. But you know what we can do, and maybe I'll talk a little about it, but we have created these mice which don't have an immune system. So we can take human tissue and we can put it into the mice, and the human tissue will grow in the mouse. But because these mice don't have an immune system, so we have genetically engineered mice to not have T cells and they not have white blood cells, basically. Yeah. And so I have a slide actually about that, maybe. Okay, that goes into cancer.
Are they difficult to maintain?
Yeah, they are very hard to maintain. So they actually live in a very clean environment. We sterilize the cage that we give them. We sterilize their food. We sterilize the water, and they get infection very easily. And then and they also they don't live very long because they don't have an immune system. So it's not great for the mouse. But actually, they have a pretty good life, because if they were in the wild, then they have to, fend for themselves, get their food here, everything, they have perfect temperature control. Everything is really, yeah.
Do they have gut bacteria, going back to last week's lecture?
They do have gut bacteria, interestingly, some amount of gut bacteria. They do have it. Yeah, I know Dr. Michael's so we have 2 rooms in the mouse facility, and my room has these very immune compromised mice is what we call them. And then Michael's has this clean room right next to it where he has, no bacteria there. Yeah, but those mice have an immune system. My mice don't have an immune system. So
But they do have gut bacteria.
I think, yes, they have gut bacteria. I have not looked for it, but I'm sure their gut bacteria are going to be very different or very minimal as compared to the gut bacteria of the mice that have an immune system. I'm sure somebody has looked at the gut microbiome in that. Yeah. So now, how does breast cancer develop, right? So what happens is this is the normal ductal structure, and then over time with just going through life, right? These cells, I was saying that, sometimes they can, because they have to replicate their genome, they can have a mistake sometimes, and a gene that is very important to tell the cell when to divide and when not to divide. And what will happen is over time, these cells will start to accumulate into the duct. And this is why, women should get mammograms. And after the age of 40, I think they recommend it every other year or every year, depending on because these cells can divide and in the duct. And if they are in the duct, that's fine. I mean, actually, early stage breast cancer is very curable. The problem happens is when they leave and they go to a different place. So usually the doctor is able to see this thing called invasive ductal carcinoma. And basically that's like what we call stage 0 of the breast cancer. It's the first step where the cell has now decided that it's going to leave this red color thing. It's going to leave this basal structure and move outside. And that's actually where, actually, even at this stage, it is pretty curable. Usually women can get a reduction mammoplasty or a lumpectomy, and that's going to be okay. So that's our breast cancer. But I don't know if folks know, but I was just going to also talk a little bit about different types of breast cancer, because people just call it breast cancer, but there are many different kinds of breast cancer. And this is sort of a very, if you go to American Cancer Society and if you just type types of breast cancer, this is what comes up. And then I wanted to, just because we are talking about breast cancer, I said, this would be a good, it's not really what I study, but a good background. So the thing about most breast cancers are these, what we call luminal A and luminal B. The reason they are called luminal A and luminal B, that's just how pathologists call it, is that because it's about how they look. The breast cancer looks like the luminal cell that was the milk producing cell. So it's not that it is the milk producing cell, but it looks very similar to the milk producing cell. And this is actually the majority of breast cancers that occur in women around, postmenopausally. The ones that are diagnosed happen to be what we call luminal ones. And they are the ones which express this estrogen receptor, progesterone receptor. And they are the ones that, they are given this drug called tamoxifen, which is going to block estrogen. And they are the most common type of breast cancer in women. It's also the one that has the best treatment outcomes in women. Then there is this other kind of breast cancer called the HER2 positive breast cancer. And HER2 is basically a gene that is expressed on the luminal cells. And sometimes these cells can make much more of this gene. And this breast cancer is much less in women, but till I think early 90s, this was not a great diagnosis because they didn't have a targeted drug. But then there are researchers in UCLA that are now able to make a drug against this protein. And so these breast cancers are also, they have very good prognosis, not as, I would say, almost similar to the luminal A and the luminal B. And then the last one is what we call triple negative breast cancer, which means it doesn't have estrogen. It won't respond to estrogen. It does not need progesterone. PR stands for progesterone receptor, estrogen. And then it does not have HER2, which means it lacks all of the three. And this is the place where a lot of young women get diagnosed with this breast cancer. These are also the women who have BRCA1. If you've heard of BRCA1, BRCA2 mutations, those women end up getting this basal-like breast cancer. So, which is why, Angelina Jolie decided to do a double mastectomy because she's a BRCA1 carrier. And the thing about this breast cancer, it's very aggressive. Usually by the time it's done, there's no targeted therapy option. So the standard of care remains chemotherapy, which is not good. And it also has a highest chance of going to a different place and becoming metastatic, which is basically when a cancer moves from the primary organ, the breast, to like somewhere else, we call that metastatic. So it has moved out and become something that is harder to treat. Any questions on this? Okay. The reason, because I think all of this then leads into what I do, but so what I study in the lab is mainly this kind of cancer. We have now started to do a little bit on this cancer because this is the cancer that, the breast cancer protection that with pregnancy, that really reduces this breast cancer. It doesn't have so much of an effect on this cancer, but this cancer is the one that is more aggressive, and right now lacks any treatment options. It's also much higher in the African American population than the Caucasian population. So there is there's a racial predisposition to this breast cancer, and this is a whole area of super active research. So yeah, I came from both my PhD and my postdoc were in very clinical labs. So I worked very closely with oncologists. So my research also, I tend to focus a lot on the clinical aspects of research. Even now in the lab, try to bring in that as well. So yeah, so I just wanted to say best prognosis to worst prognosis. Best prognosis just means the outcome of the breast cancer. Yeah.
You said Afro-American, how about in Africa?
So yes, even in Africa,
Okay.
there are more. So what people have done, there's this amazing group. What they did is they actually went to Africa, sub-Saharan Africa, and they got samples from women, and they did sequencing to figure out the DNA, like what is there. And then they sequenced women in the United States, and they looked at the ancestry, and they look at like what are the genes that are actually that's changing similarly or differently. And they find that a lot of genes in DNA repair, which means like if our DNA by mistake breaks and it needs some proteins to bring it together, those are the ones that are changing. And not only that, people are now doing like, interestingly, even in India, there is more prevalence of basal-like breast cancer as compared to the luminal. So we don't understand fully what is it different about our genomes, because we all are different, that drives certain cancers. So for example, another form, like I know I'm talking breast cancer, so but colon cancer was my first love, right, in some ways. But like, so colon cancer is very prevalent in the United States, but gastric cancer is very prevalent in Japan. So it's the same, if you think of it, it's our same digestive system, but just different places. And then we don't see as much of liver cancer in the United States, but liver cancer, again, in Asia is very, it's much more prevalent as compared to here. So there is definitely some genetics behind what's like, not genetics, but depending on genetics and environmental factors. I think both contribute to like these cancer risk. Yeah.
I think aren't a number of plastics like BPA, BPE, estrogen inhibitors? How much of an effect is that having? Because we're all full of.
I know. Yeah. I don't know the numbers, but it does have an effect on cancer. Like I don't know what contribution exactly. So I can look up the percentage. Yeah. Yeah.
Can you repeat the question?
The question was basically the microplastics that are there. and also carcinogens that occur in certain plastics. We've eliminated a lot, like now it's all BPA-free plastics. But yeah, I know PP also, but I don't know the numbers on that. Yeah. I know the lung cancer numbers, which are also very bad with the tobacco also, and that affects, cancer risk as well. But yeah.
I recall that there was within Africa, sub-Saharan Africa, there's more between group genetic variation than there is anywhere else. Is the sequencing differentiated by the different?
Yeah, they are doing it. They just, there is this consortium where many people come together, and then they have just got a huge amount of funding recently to actually be able to look at all the genetic differences between those different ethnic groups, and also be able to trace like what ethnic groups came here. And it kind of goes back hundreds of years, right? So yeah, people are doing that. So, UCSC is the home to the genome browser, where the first human genome was sequenced, but our first human genome that was sequenced was very basic. We didn't have all the information. So now people are sequencing across like multiple things. They are sequencing so many tumors from people. So we are getting just much, much more information on what's going on. But we don't fully understand the implications of that. All we are doing is collecting tons of information. Yeah.
Maybe you can correlate some of it sometime.
Yes, and that is like the ongoing work, but it's going to take years because yeah, we've created databases and databases of information, but now we have to test out and do the correlations and yeah. And then I just wanted to also just talk about different stages. So basically, when we talk about cancer, so my lab mainly focuses on this metastatic disease. Metastatic disease is when the cancer has moved from the breast to a different place. This happens to be the most challenging to treat patients because we can't, for the most part, once a woman is done, and I don't say this lightly, I understand like a lot of implications to it, but for the most part, once a woman is done with the mammary, with her birth years and she is given, we don't need the mammary gland as an organ to survive. We need it for other things like, but we can take out the tumor and be able to treat it. It's much harder if the tumor is now gone to the brain or the bone to be able to then treat it, because there is no way for us to get in. There are ways to get into the brain, but that leads to it's much more difficult than being able to get a tumor out from the breast. And that's primarily like where my funding is on metastatic disease. Okay, and so I'm going to now talk about so we have stem cells in the mammary gland which give rise to these cells which are going to produce the milk and push the milk out. But what happens is, what I was trying to emphasize, is these stem cells are sort of the long-lived cells in our body, right? So if you think of cancer, which occurs over decades sometimes, that is not occurring from these cells most of the time, because these cells are going to die after, quickly. Even in the intestine, the cells that absorb our food, they are going to be gone. Like every week, they are gone, right? So the mutations that usually occur in these, what we call are the stem and progenitor cells, because they are the long-lived cells in the body. So what I study in the lab is like how these stem cells change as we age. So we have young mice, and then we have mice that are 18 months, so the young mice equate to like between 20 to 30 years. And then we have mice in the lab, which are 18 months, which equates to 56 to 63 years. And so we then study how these stem cells are changing with age, and what does that lead to in terms of cancer. So that's one project in the lab besides the metastasis project also. I know is there a time that,
Well, I mean, we kind of go till noon. so-
okay, all right, okay. So now I'm going to introduce this concept of what is tumor initiating cells. Basically, initiating means the starting cells of the tumor or the ones that have the capacity to form a tumor. And so these stem cells over many years can get the right kind of mutations, which is a very low probability. I just want to emphasize that it's a very, very low probability, but just remember we live for a long time. Our cells are dividing and these stem cells are around for a long time, that they give rise to these tumor initiating cells, which can actually then form a tumor. And there is a specific reason why I colored each of those tumor cells differently. So even that is a tumor, I want to say that, not all tumor cells are the same. They are not like the M&Ms. They are actually like jelly beans. Because the reason they are like jelly beans is that not every cell in the tumor has even though it all came, they all have the same mutations and they all are part of the tumor. They are still very different from each other, even in what they can do. If that makes sense, right? So not if we've done experiments where we can take different cells and put them in mice, and not all of them from the same tumor, depending on we can use different ways to put different cells in the mice, and not all of them will give rise to a tumor, even if they have the same mutations. Which is interesting if you think of it from a therapeutic standpoint. Chemotherapy does not destroy all cells. Some cells get left behind. Why do those cells get left behind? And that's probably because not all the cells in the tumor are the same. If we were able to efficiently kill, if everything was exactly the same, they all should have been killed by chemotherapy. But it doesn't happen. And so that's why we know that our cancer cells, just like cells in our body, or even if they are all in the tumor, they are more like jelly beans and not MM. And so basically, in therapy, then that's why you have relapse occurring. So
This specific to breast cancer cells?
no, it's true for all cancers. It's true for glioblastoma. It's true for lung cancer. It's true for lung cancer, colon cancer, which is where I actually started working with this whole concept of stem cells. And then it's true. There are some conflicting reports about melanoma, that melanoma may not follow exactly this pattern, but melanoma is a very aggressive disease to begin with. So, by the time, till very recently, we didn't have good therapies for stage 4 melanoma patients. But now that's changing. But yeah, it's almost all cancers are like that. Leukemias, they are all follow a similar pattern. Yeah.
And cells continue to mutate as they grow, right?
Yes,
As you make more and more cells. are
Yes.
born. How about immunotherapy?
Yes, that's a great
Yeah.
question. So immunotherapy, right? So what happens is so if you think about immunotherapy, the way it works is, I don't know if everybody knows exactly what immunotherapy is, but the way it works is our immune system is kept in a check, because if we have a hyperactive immune system, it's going to destroy our own tissue. So in our immune cells, we have basically two kinds of. So this is going to be a little different than the thing, but that's okay. So we have our blood stem cells. And they give rise to two kinds of cells. One will be, we call them myeloid. I mean, fancy word for basically saying these are going to be all giving rise to RBCs and some other cells over here, which I'm not going to label. But this is what we call, this is the lymphoid. which basically gives rise to two kinds of cells, the B cells. These are the cells that make antibodies in our, when we get a vaccine. And then the other cells are the T cells, which will give rise to, which will basically, their job is to go and kill a cell that is infected with a virus or something. So this will, the T cell can go and kill other cells. They can also respond in other ways to infection. So that's why when we get a vaccine, in the lymph node next there you have a lot of pain and inflammation occurring, because basically what's happening is those T cells and B cells are getting activated. B cells are mostly in the spleen, but the T cells are in the lymph nodes, they are circulating. So we have lymph nodes all over our body, which are sort of the ones that, all the fluid from our different tissues is draining over there, and they are checking constantly whether the cell is getting infected or not. So, but it's kind of cool that our T cells don't attack our own cells, right? And so during development, we go through this process where any cell that attacks our own cell is told like, okay, you're not going to be around, you need to die because you can't attack. And then if you don't have that process going on correctly, we end up with autoimmune diseases. Or even sometimes later in life, people come up, have autoimmune diseases. So the way immunotherapy works is that our T cells are kept in extremely tight controls. So much so that even though they know that there is a cancer cell, the cancer cell can do things to hide itself. So it can pretend that, it can pretend that, I'm a normal cell. Don't pay attention to this mutation. Don't kill me. And then what was discovered is that if we remove that block on the immune system and activate it by using these immunotherapy drugs, we can actually take that immune cell to go and be able to kill the cancer cell. And that's actually, James Allison, who got a Nobel Prize for immunotherapy in 2018, that's what he discovered together with another scientist whose name I'm forgetting, from Japan. And so they found that there are these what we call checkpoints in the immune system. And if we block those checkpoints, we can leverage the immune system to go and attack the cancer cells. And that has literally changed the outcomes for melanoma patients. So when James Allison, when he gives his talk, he says, the first time I talked to people about this, people left the room. They thought, it's not going to work. And then he talks about this woman who was diagnosed with stage 4 melanoma. She was 22 years old, and she had no hope. And then she was the first patient to receive immunotherapy. And then, she went on to having kids who are teenagers. So it was like, it doesn't work for every cancer the same way, and it doesn't work for everybody the same way. So we are still trying to understand why is it that some patients benefit from immunotherapy and some patients don't. So we are trying to understand the molecular basis of what's going on between the immune system and the cancer cells. And different cancers. So each cancer. So for example, since we are talking about immunotherapy, so I told you triple negative breast cancer does not have a targeted treatment option and affects younger women. They are just finishing up the clinical trials for doing immunotherapy with chemotherapy for triple negative breast cancer, and it is showing promising results. Not to the point that it's going to be a full cure, but it's better than just chemotherapy. So yeah. Okay. Okay, and so we also something that we do in the lab, which I wanted to introduce, is this other technique. So I spent most of my graduate and postdoctoral career doing is waiting outside the surgery room. So we want to study human tumors, right? How do we do that? We can't like start doing experiments with humans. So I talked to you already that we have these immune compromised mice where we can grow these human tumors. So what we can do is when a woman or any patient from any surgery, it doesn't matter if it's breast cancer, but we can get a small tiny piece of the tumor and we can put it into our immune compromised mouse, and then it will grow in the immune compromised mouse. So this is the mouse, and you can see that I have implanted the tumor here, and this tumor that came from a patient is growing in the mouse. And then now we have a source for being able to study human breast cancer, or it doesn't matter, human colon cancer, brain cancer, in a mouse setting. And we can then take these tumors out and actually, have them in a dish. We can chop them up, we can get the cells from the tumor, and we can actually then start to study whether those cells are like M&Ms or jelly beans. So, but to do it from the person undergoing surgery is really, really hard, because the first priority for the surgeon is to take the tumor out and send it to the pathologist for doing the tests, whether what kind of therapy should this person receive, and also like all other kinds of things. So what we get in research is the last bit that they don't need in the end. And of course, the very important thing for all of this is consent. So we have to consent the people undergoing the surgery that, would you be willing to donate it for our research? What are you okay with? What are you not okay with? Because previously, back in the day, there was no way of informed consent, and there were a lot of things that happened that were not right. So if people have read the book about Henrietta Lacks, and she had cervical cancer and her cells were taken, they were grown. It's fine. I think like the people who did it with the intention of research, but then it was commercialized, and then the company started selling that, and that was not the right thing. So anyway, very digressing from everything, but I do think this is important in terms of how research is done. So as I said, we can make them into single cells. So each tiny dot over here is a cell that came from this tumor. And these cells are very, very close to how they are in the patient that they came from. So people have done experiments where they have given the patient the same therapy, and they have given it to the mouse, and they have looked at what happens to the mouse and the patient at the same time. And they have been able to predict what will happen based on the mouse studies. So it really helps us understand how to do the research. But as you can tell already, these mice are super hard to maintain. They are also, mouse costs are a big thing in research. So I pay a lot more to maintain these mice in here at Santa Cruz. My mouse bill per month is $3,500. And that is doing no experiments. That is just to keep them happy. That's it. But so we have to come up with better ways. Also, this is really not great for us to be able to screen drugs, right? Because we want to test a whole bunch of drugs on a whole bunch of kinds of tumors. We don't want to study just one patient tumor. We want to do it across multiple different tumor lines, multiple different subtypes of breast cancer, multiple different ethnicities. So the numbers can quickly grow to a thing which is unmanageable. So the other system which we use in the lab is, we call this the breast organoid system. And I think like it's okay if we don't get through the rest, but the system is so cool. So what we can grow the cells in a dish. People who have been growing cells on a plastic dish for like years. But again, that's not a great way to grow, because our plastic is not how our body is. The tension that is there on plastic, the Pascal that they call to measure like the force, is very different on plastic versus our the tissue, the fatty tissue in our breast. So how will the cells grow the same as they grow inside? So after many, many years of research, what people have found is this gel that is that we can obtain from other cells, and we can grow these breast cells coming from either the patient or the tumor or from the mouse, and we can grow them in a 3D structure. And you can ignore all the details over here, but basically we can grow them in a 3D structure, and they will look like very similar. They will form the same kinds of structures. They will also look very similar to to the mammary gland in vivo or in the breast, how the mammary gland looks. And the cool thing is we can add hormones and we can also produce milk in vitro. So this is our in vitro milk production system. And the milk, it looks, we don't even sometimes need to stain the milk. If we look under the microscope, we can see, and if we put it under a fluorescent light, milk glows green in color. So we can just see the milk. Pretty cool. Yeah. Yeah. Okay. I think I can stop here if it's, or yeah, because I get into then a whole other thing. Maybe one other technique which has changed the way we do research, if that's okay with you folks. Okay, this is what I'm going to skip all of this. And so I think let's skip this. Okay, so, we know that our cells express DNA, which means it has all the DNA has everything that can be made, and all the RNA, which will be what we make. Like, so we can take a recipe from the book, the whole cookbook, to make what we want to make. And every cell in our body makes different things, right? So our liver cells make the things required for the liver, or our retinal cells make the pigments that allow us to see. But every cell in our body has the DNA to actually make everything. And that is what actually, we don't think about it, but the cancer cell can do everything. It is smart enough because it has the whole cookbook. It can do what it wants in some ways, which is why the cancer cells can go to the brain and start growing there, because it has become mutated that it is doing things it's not supposed to do, but it has the recipe to do everything, because we all came from a single cell. And yeah, so many of these cancer cells, right, they can hide from the immune system. And normally they're not supposed to hide from the immune system, but they can do it because they know what gene to express, or they become changed enough that they can express this gene, which will make them hide from the immune system.
But the mutation rate in a lot of cancers increase tremendously as they sort of lose control.
Yes,
Yeah.
absolutely. Yeah, and so that the mutation rate, so they lose this one gene, which is we call the guardian of the genome. It's called P53. Such an exciting name, right? Because they found the gene was running at 53 on the thing, so they called it P53. But it was called the protein of the year. I think it made it to a magazine also at some point, got a front cover, and it's the guardian of the genome. And once cancer cells lose P53, they have a lot more mutations, because there is no way for the cell to know that it has a mutation and I need to stop. So most cancers will 50% of human cancers, 50 to 60% of human cancers would have lost P53, and the remaining cancers in some way have no function of this gene P53, which then, there's another thing, right? Like, why do immunotherapies work for certain cancers and don't? Interestingly, the cancers that are more mutated, immunotherapies work better, because those cells become so different, because there are so many mutations, that the immune system is able to see them. So 2 years ago, there was on the New York Times, they were able to use immunotherapy for this one kind of cancer and cure the patients. It was an article in New York Times where these people who are born with defects in this gene involved in DNA repair, they, for the first time in their lives, they would actually be cured of the cancer. And those patients get cancer very early, like early in their 30s. They get colon cancer. Yeah, so all of these concepts are very intertwined. Cancer is like, it's studying everything, basically, and everything that goes wrong in the cell. And in fact, a lot of things that we know in normal biology have come from cancer biology, because people started studying cancer and trying to cure cancer, and then they were discovering things that were needed for the normal cells. Because the thing about these cancer cells is that they grow, so we have been able to grow them in the lab. So almost every lab, will have like some cells growing, which in some way are not are not fully normal, because our normal cells, they can't grow forever. They're not supposed to grow forever. But these cancer cells can grow forever. So labs use them because it's easy to grow them in the lab. So, yeah. And so, yeah, then we can make the protein, and then this protein can, give rise to different cells and tissues. So there is this very new technology which has really changed the way that I do my research. So remember I said that the cancer cells are like, are they like M&Ms or jelly beans? But there's jelly beans, right? But how did I even find out that they were like jelly beans? And how did all the researchers find out? So what we need to do is be able to take the RNA that is in the cell, which is right here, to understand what the cell is going to make, right? So there are ways in which we can take this RNA and look at it and say, what is the cell making? But for a long time, we had to take all the cells over here and get all the RNA. And then, we could never tell whether it was like as if instead of taking one jelly bean and eating at a time, I took a bunch of jelly beans, stuck it in my mouth and said, okay, this all tastes the same. There was no way for me to take individual jelly beans and taste them. So for a long time, and this is a complex slide, and I'll stop here after this, is that there was no way for me to tell what this green cell is doing versus the red cell. It was like also drinking a smoothie, right? I was like a mixture of everything just blended in. It tastes really good, but I can't tell how many strawberries or bananas are in there. But now what we can do is we can actually we can look at one cell at a time, and we can taste each jelly bean and see exactly what it's doing, or eat like a fruit salad. So we know how many grapes are there, how many things are there. And that is what has told us that our tumor cells are not like, they are not all the same. They are like jelly beans, because we've been able to actually, with a lot of computational biology, with a lot of technology development, and all of these are really complex terms. I apologize. I should have simplified them. But the idea is that because of the single cell is why we've been able to look at whether they are jelly beans, M&Ms or not. Yeah.
That's amazing.
Yeah, it was very cool. And it was developed, in the Bay Area, I heard. So, and San Diego. So still all California. Yeah. Yeah, I'll stop here and, see if people want to chat or do other, talk. So if there are any other questions, happy to talk about it.
Yeah, since you mentioned the P53, do epigenetics play a part?
Yes, it does. Yeah. So there are these different, we call them the hallmarks of cancer, right? So the first thing that a cell needs to do to becoming cancerous is that it needs to remove. So cancer is like a car. The first steps for a cancer is like a car. The brakes are not working and the accelerator is stuck. So the brakes are like P53. So P53 is a brake of the car and it's not working anymore. So it can't tell the car to stop. And the accelerator is like genes that are involved in making the cell divide. Now, that is the first step, but that is never enough to get cancer. Or not the cancer that we see in, it's like the first step. And then what we need is other kinds of changes to become metastatic. And interestingly, what research has shown is that if you take the tumor from the primary site, so say take the tumor from the breast versus and the same patient, so no genetic differences, you take the tumor from the breast versus tumor from the brain, which is basically primary tumor, and we call that a metastatic tumor, and you sequence them, you don't see more mutations in them. The changes are all epigenetic. So the changes are occurring not in the DNA sequence, but they are occurring on the DNA through these other changes or changes in genes that are modifying the way that the cell can go from here to there. Yes.
So have they looked at some of the drugs that remove?
Yes, they are, and some of them have made it to clinical trials. I'm trying to think the one that is, there's this one, I don't know at the top of my head if they are approved for, but they are being testing these epigenetic modifiers in clinical trials for patients, yeah.
Yeah, imipramine is one.
Which one?
Imipramine, I think it's one of.
Okay, I don't,
There's two.
okay. Yeah, I looked that up. I'm more aware of like breast cancer, so I don't know if it's, yeah. Yeah, but I know of a couple that at least went through phase 3, so they should be approved and being used, yeah.
In a metastatic tumor, they could do a DNA sequence and determine where it originated, right? So that you know it's a liver cancer, it's a breast cancer.
Yes, in most cases, they don't even need to do that. The pathologist can look at it under the scope and be able to tell where it came from. Yeah, and then in some rare cases, they can't tell, but those are extremely rare cases. Yeah.
I always wondered, when someone says that already, they died of liver cancer that originated from breast cancer. How do you know that? Now I know.
Yeah, it's the pathologist, it's amazing how our cells actually look so different under the microscope. And I'm not a trained pathologist, but I know a little bit about looking at cells, but a trained pathologist can tell you exactly where that cell is coming from and whether it's a liver cell, a brain cell, or it's a cancer cell or a normal cell.
And so can AI virtually.
And eventually, I think it might be able to. Yeah, but we'd have to train it over a lot of models. Yeah. Similar things for radiology, right? That's another thing where AI can be able to, yeah. I don't know. I feel like humans are still needed at the end of the day, but we'll see how that will change. Yeah. We use, a lot of our so a lot of computational work that we do is based on some of the neural network and large, not exactly not the models that are used for the AI like ChatGPT, but we have like models where we train the model to look at cells and sequence like the single cell data. We have them iteratively look at those cells and then be able to predict which are the tumor initiating cells or which ones. And that was the rest of my talk is we can do that. And then by looking at lots and lots of data, it's able to say, you know what, this is going to be your stem cell. This is going to be the tumor initiating cell by just looking at gene expression. And that's kind of cool because then we can look at what genes are in those cells and then study them in the lab. And that's primarily what I do. Yeah.
All right, so we should probably wrap it up there. Thank you very much for being here.
You're a very good teacher.
Thank you so much.